All right. Good morning, everybody. Thank you for surviving the first evening of revelry of the Longevity Summit Dublin 2023. Of course, there will be subsequent mornings of revelry, so anyone who feels that they're already short of sleep, get used to it. It's not going to change for the next until Sunday or Monday. But of course, what I have done in order to ensure that you have no excuse for catching up on sleep is to organize particularly explosively wonderful sessions at the beginning of the day each day, and today is undoubtedly no exception. We have a session now comprising three of the absolute most prestigious people in the biology of aging worldwide that you could possibly imagine, and I am delighted to be chairing this session and moderating the panel that we're going to have at the end, where gratifyingly only 5 minutes late getting going. I hope that we will be able to keep somewhat on time today, but we'll see whether we actually can. In the meantime, though, let us begin. The first talk of the day is my friend Vadim Gladyshev, who will now come and tell us all about what he's been doing in the past little while, which is honestly pretty amazing, but judge for yourself.
Good morning. Well, first, thank you very much for the kind invitation. So I always enjoy these meetings. And when I read in the program, I can see this title, The Elusive Concept of Biological Age, which I really liked. So I just put it as a title of my talk. So then, what is biological age? So it's kind of, when we think about this, is somewhat obvious. So some people might age more rapidly, some slower, and this kind of reflects those changes in biological age compared to the average person. However, when we think more deeply about this, actually becomes not as easy to define. And the reason is that it's a kind of link to the concept of aging. And aging itself is a very kind of questionable term, because we use this to refer to completely different processes. And this is like this. So some of us think of aging as an increased chance of dying. Some demographers, for example, think about this. And some other people, like more kind of evolutionary people, think more of fitness, kind of decline in fitness, or maybe functional decline. Some people that use physiology. So molecular people might, for example, think of damage accumulation, or like developmental people might think of continuation of development. And of course, later in life, all of these processes go hand in hand. However, they are not the same. So what actually aging is, there's no consensus currently in the field. And this is critical because it really depends on how we approach this question. How do we, which methods we use to target it. And so this is how I think about, in a very simple kind of way, about this process. So my thinking is that aging kind of begins from molecular things, damage, basically damage in a broader sense, not just kind of byproducts of metabolism or mutation, but kind of any negative consequence of being alive, because we have all kind of genes and processes, they do whatever they do, but at the same time, they also produce kind of unavoidable negative consequences, and these consequences accumulate. And eventually, they result in functional decline, diseases, and finally mortality. However, mortality may be also caused by other features. You know, it's a very complex trait, and in fact, the diseases as well. So my thinking is kind of via on the left here on the damage side, and then it eventually results in all other manifestations. However, those manifestations are also influenced by other processes. So to kind of begin addressing these questions of what is like aging, what is biological age, how can we define, how we can target it, we recently formed a consortium called Biomarkers of Aging Consortium. And the first manuscript, which is shown here, actually will be published later this month already. And this is a group of about 25 different labs, many of them leading experts in the field, including actually all three panelists in this morning session. And it was kind of headed by these three people on the top. It's Madi Mokri, who's a postdoctoral fellow between my lab and Mike Snyder in Stanford, and Chiara Herzog, who is in Austria, and Jesse Paganik, who is also a postdoc in my lab. And so this consortium kind of met and over the year kind of discussed all of this concept and resulting in some kind of consensus, which is not the final truth, of course. It's just kind of current state of knowledge, at least agreed by this community. Also, I'd like to mention that actually this group would organize a first symposium on aging biomarkers, specifically focused on aging biomarkers, which is going to be December 4th at the Bach Institute. So let's see what this group came up with. And these are some of the definitions that we put in the paper, in the first paper. And you can see, like, what is aging? So aging would be, in a simple way, could be thought of a process of accumulation of consequences of life, such as molecular and cellular damage, that leads eventually to functional decline, chronic diseases, and ultimately mortality. So what is healthspan? Healthspan is a period of life prior to onset of chronic disease and disabilities of aging. In other words, the organism spending time in good health. What is biomarker of aging? It's a quantitative parameter of an organism that either alone or in combination predicts biological age and ideally its changes in response to interventions. So biological age, that's actually a topic of this panel, this session. Conceptually, it would be individual age defined by the level of age-related, age-dependent biological changes, such as molecular and cellular damage accumulation. Of course, in practice, we refer to biological age in a population, in reference population. So it would be a number matching a chronological age where an average person in a reference population shares the individual level of age-dependent biological changes. So we always think of reference population when we discuss biological age. I will skip chronological age, which is obvious. And another concept that is currently actively used in the field is so-called age acceleration, which is a difference between biological age and chronological age. And in this manuscript, we actually proposed instead to use age deviation, or age-dev, to distinguish it from increased rate of aging and also to encompass kind of changes in both directions, because it not only can be accelerated, but in a way decelerated. Yeah. And finally, what is geroprotector? That's another key term which is not going to be discussed, I think, today, but that's another also important concept, which is agent or some kind of intervention that increases health span or lifespan and ameliorates biomarker of aging. So, of course, this is a very broad and maybe in the future can be defined in a better way. Yeah. So biomarkers, of course, they're different. These are different parameters. There could be different biomarkers of aging, and these are kind of also figure from the paper where we define types of biomarkers. They could be molecular biomarkers, for example, based on omics approaches, kind of on the left-hand side, and you can see omics like genomic or transcriptomic, proteomic, metabolomic. Of course, mostly we apply epigenetic clocks, but in fact it could be any omics approach. There are also physiological biomarkers of aging. This would be measuring something that measuring functional performance and also physical characteristics. And also could be digital, for example, wearables, but also non-wearable kind of technologies that can be used for this purpose. And on the other dimension of biomarkers of aging, we could define them as predictive biomarkers. For example, tools that could predict, for example, future mortality or kind of future kind of consequences, functional decline, age-associated changes, and so on. Also could be biomarkers that so-called response biomarkers. This is something that the parameter that reports the changes in response to some kind of intervention or changes in the environment, for example. And also discovery biomarkers. that tools that could be used, for example, to discover future drugs, future longevity interventions. Now I'd like to show a few examples from my lab, recent examples where we kind of apply these biomarkers to define biological age, and there's no kind of ideal kind of a study, so I will just review a few that kind of addressing this from a different perspectives. So this slide shows our first clock. It's a mouse clock. We already published quite a few years ago, where we apply this to interventions in mice. And on the left, you can see a blue line. These are 16 age groups of mice, and on the x-axis is the actual age, and the y-axis predicted age, and you can see there is very good correspondence. And then we have 4 groups of mice subjected to calorie restriction in red, and you can see that all of the 4 tested chronological ages, they are predicted to be younger. We also know they live longer, and they're younger, kind of all this makes sense. So this biomarker, even though it was developed for kind of chronological age, already can kind of report changes in biological age. In the middle, it's another intervention. It's a genetic intervention where we removed a gene for growth hormone receptor. And the mice on the left and right, they're chronologically identical mice, but then the knockout mice, growth hormone receptor knockout mice, they're predicted to be younger. And on the right side, we collect fibroblasts and then convert them to induced pluripotent stem cells, which correspond to the embryonic state. And you can see that this same biomarker predicted in the mouse blood actually can also report reduction in biological age all the way to embryonic state, which is approximately 0. This is another example where we developed a single cell clock. This is epigenetic clock that quantifies age, predict age in individual cells. So, for example, it's just one example of that, where we each dot corresponds to one cell. On the right, it's hepatocytes from a 26-month-old mouse, and you can see individual cells correspond approximately to the dashed line, which is the actual age of the mouse. And in the middle, these young hepatocytes from a 4-month-old mouse. It also kind of reports approximately the age of individual cells, although it also reports heterogeneity. And on the left is a mouse embryonic fibroblast. Embryonic cells, they're younger, and they're also predicted to be younger based on this clock. The advantage of this clock is also that we could predict the age based on a relatively few number of sequencing reads. Sometimes even like 10,000 reads could be enough to predict the age using this tool called SCH, which stands for single cell aging clock. So what emerges from this is that cells in the tissue, even though they talk to each other and approximately coordinate their age, but at the same time they also have differences in the predicted age. It seems that some cells age a little bit faster than other cells, certain senescent cells, for example, and some cells age slower. Like we find certain stem cells, they age slower. And in the end, it's actually quite significant heterogeneity in the tissue. So when we always think like of biological age of kind of uniform across tissues, but actually it's not. It varies across organs, across different cells, although, of course, as I said, there is also close interaction among those components. Okay, I'd like to give this example, the example of the organism called naked mole rat. And the interesting thing about this organism is that it doesn't show increased mortality with age. The mortality rate actually is quite stable, at least in the period from maybe like 0 to 15 years old for these animals, which is shown on the right. So the question is like whether this organism actually ages. And in the literature, it's often referred as a non-aging organism simply because it doesn't show increased mortality with age. So we wanted to check whether this is the case, and so we developed an epigenetic clock for this organism. And actually, it can be built very well, and we could predict the age of these animals. There are 2 papers. You can see it's Csaba Kiripeshi, who's a postdoctoral fellow in my lab, published one paper. Another paper by Steve Horvath and Vera Gorbunova. Actually, both papers came up with more or less the same conclusion, that the age can be predicted. This means, at least epigenetically, this organism ages. but demographically it does not. So, of course, this is it's kind of paradoxical whether it ages or not. In my mind, it does age, even though it doesn't show increased mortality rate with age. In fact, we could actually quantify the rate of aging of this organism, because here we compare kind of NECMO rats, mice, and humans. And it's a little complicated slide. Maybe you can see in the middle on the left. In green, it's kind of human changes. In blue, NECMO rat changes. In orange, it's mouse changes. You can see that NECMO rat is kind of in between mouse and human. It does seem to age slower than mouse, but faster than human, but doesn't show changes in mortality. So this is another example published a few months ago, and here we tested the effect of stress, severe stress. So what we find is that under conditions of severe stress, but not mild stress, so biological age can be rapidly increased. And once the stress is relieved, biological age returns to approximately normal state. And we describe 4 examples here: major surgery, severe COVID-19 infection. It's not just infection, but when somebody actually gets to a hospital, to ICU, on ventilator, and then biological age is maximal at this point. Once the person recovers, actually the biological age is predicted to be lower again. Another example is pregnancy. Women in the 3rd trimester seem to be a little bit older, and then the biological age is reduced postpartum. And the 4th example we describe is parabiosis in mice. It's a heterochronic parabiosis when we connect young and old mice. And in this case, the younger mice in these pairs, they increase biological age. Once the mice are separated, it also recovers. Of course, the question here is that whether, there are 2 questions, whether we actually perturb biological age here, or which is some other parameter, like inflammation or some kind of immune response or whatever, cell type composition. So again, I think it's important to think about the nature of aging. So what is aging? In my mind, it's a kind of cumulative, deleterious consequence of life. And it's just these consequences, they accumulate under severe stress. More damage is produced. And in a way, it kind of increases biological age. It manifests as various parameters, including cell type composition and immune response and so on. And once this is released, organism is able to deal with this damage, and it's reduced, and also biological age is reduced. Although we actually don't call this rejuvenation, because that's another term that is kind of not very well defined, because what is rejuvenation? It's obviously a reduction in biological age. But then the question is whether it should be a reduction over like artificially increased level or a reduction relative to the normal level. So in this paper, we actually use age reduction instead of rejuvenation. But in the paper which is shown on this slide, we actually do use the term rejuvenation. And here is the work by Bohan Zheng, who is recently graduated with PhD in my lab, but it's a collaboration with Jim White and Steve Horvath and other labs. Paper published actually about 2 weeks ago in Nature Aging. So we here study the effect of heterochronic parabiosis on old mice. We connect the mice so that they develop a joint circulation and keep them for 3 months. And after 3 months, we separate and follow them for another 2 months, measure biological age, and then follow them until they die. So what we found on the left is that the lifespan is increased of these mice. So after young and old mice are connected, it seems the younger mice, they rejuvenate old mice. And 2 months later, actually they're still younger than controls. And eventually, they also live longer. On the right side, here you can see that it's a predicted epigenetic age based on, in this case, we used reduced representation by sulfide sequencing, but also we have data on the Steve's kind of clock foundation, which is the Illumina array, and also transcriptomic age. They all show the same, basically, that the age is reduced, and then 2 months later, it's still lower than control. Although it seems like over time, this effect diminishes. So in the end, we see that the mice live longer, but the effect is not huge. So it seems like this effect kind of disappears over time, even though it still remains to make them longer lived. So this is another example where we apply clocks. And here we use clocks that we developed for adult life. and apply them to early embryonic development. First, like a big realization in the field, at least for me, is that when we develop the clocks for the delta ages and apply them early in life, we find they still tick. So if, for example, if we apply like 20-year-old, they tick, 15, 10, 5, 1, they still tick in the same way. Basically, and it just immediately kind of questions this whole body of literature from like evolutionary biologists when they discuss that the aging kind of begins after completion of development, because we can see that the clocks tick early on in the same way they tick in the adult state. However, interestingly, they don't start at the conception. So we find that on the very left here, at the zygote, which is like fertilized egg, the predicted biological age is not 0. It's actually very low, but slightly above 0. And then during early development, it is reduced. And to the lowest point, we call it the ground zero state. And corresponds approximately to the stage of gastrulation in mice, approximately 7 or 8 days. And then, at this point, the clocks show that the age begins to grow. That's where we think biological aging of the organism begins. At least the current data suggest this is the case. And we published several papers on this, Science Advances paper in mice. There's another paper which is now in press in kind of human case where we find in human models. Also, we have a bioarchive paper about frogs. They all kind of point to the same kind of idea, but still, of course, this needs to be further validated. This is still kind of in a hypothesis state, I would say. So we also applied our single-cell clock to this model, and we found that on the left, the epiblast actually reduces biological age. This is in mice, 4.5 days embryonic development, 5.5, 6.5, 7.5. You see a reduction. And we also tested in 3 germ layers, endoderm, mesoderm, ectoderm. They all seem to be rejuvenated at the single-cell level. However, if we look at extra-embryonic tissues, for example, cells that later become placenta, there's no full reduction in biological age. It's predicted to be a little bit above 0, I would say. It's still very low, but not as low as in the case of cells that become the embryo itself. So just a couple of examples that we also developed a transcriptomic clock. So the epigenetic clock is not the only kind of tool available. Although it's currently the most robust and the best, transcriptomic clocks, they also work. In this case, the clock developed by Alex Tyszkowski, a member of my lab, based on several thousand samples. We sequenced most of the samples in the ITP program, this intervention testing program, mouse testing interventions, and developed clock based on this kind of lifespan adjusted measures. And so we think it actually predicts biological age of organisms. And we have clocks for mice, mouse rats, mouse rat human clock, and multi-tissue clock, and so on. And this is just one example here where we apply transcriptomic clock to predict changes in the biological age during early development, like I showed you for the epigenetic clock, and you can see that actually it's also a reduction. So it nicely kind of validates the epigenetic changes. And this is yet another example where the paper is under review currently, but there is a version in bioarchive where we apply chemical reprogramming. So everybody knows about kind of Yamanaka type reprogramming where we express OSKM factors, but also cells can be partially reprogrammed chemically. And this is a combination of 7 different compounds, and where we apply this to fibroblasts, and we can see that the predicted biological age is reduced. On the upper part, it's a transcriptomic clock. And you can see that 4-month-old fibroblasts, 20-month-old fibroblasts, and you can see that 7C, which is shown in green, it's the 7 compounds, they show a biological age reduction. Transcriptomically, on the lower part, it's application of various clocks, and in the same way, we can see biological age reduction upon this partial reprogramming through chemical means. So finally, I'd like to kind of make one particular point here is that actually challenge the concept of biomarkers, because it's commonly assumed that these tools are very good, but in fact, the reason we formed this consortium is that it's not. There are many challenges associated with the use of the clocks, and particularly when applied to interventions. In fact, I would state that actually we don't know which clock to use. We should use to test for longevity interventions, because we simply don't know. And one kind of challenge is shown here. So when we look at the same person, this is the same person in the young and older state, and when we kind of consider changes that happening for this person, you can see that there's increased risk, increased use of glasses and hearing aid, and various other features, right? And so we could develop a clock like this based on the quantifying the number of visits to doctors, like with a person in a wheelchair, all kinds of changes like this. But of course, these changes, they happen with age, but they are not aging. So they quantify aging, but they are not aging per se. In fact, in a way, they kind of responses to aging, because the reason, for example, hearing aid is used is kind of to respond to changes in hearing, right? At the molecular level, we observe the same things. So basically, some changes, molecular changes, happen in response to aging. And because they show age-related patterns, when we build clocks and apply machine learning approaches, they end up in the clocks. So basically, the clocks include changes which represent aging, some kind of neutral changes which do not represent anything, and also changes in response to aging. And so when we apply interventions, we don't know which component of the clock we target. So, for example, if we target this adaptation to aging, so in a way would be very misleading. Clocks would show that the age is reduced, but in fact it's not. So we wanted to address that particular point, and Albert Ng is a graduate student in the lab, developed the first clock we call causal clock. The paper is still under review, but again, so there's a bioarchive version, early version of the paper, where he developed a clock called cause age, and on the upper part, this is basically sites which are causal to aging, both kind of showing a reduction in aging and also kind of protection against it. And then on the upper part, like DEM age, is the clock which is built based on the CPG sites which are causal to aging, basically damaging sites. And you can see that age can be predicted very well. And on the upper right corner, adapt age. This is a clock based on changes which represent responses to aging, adaptive kind of protective changes. Also, clock can be developed like this. But of course, it's not aging, it's a response to aging. And this is well illustrated, the kind of discrepancy among these clocks, when on the lower part, we apply this to reprogramming. We reprogram cells to iPS, it's a human case, reprogrammed to iPS cells. And you can see that the damage-based clock predicts age reduction, but adaptive changes do not predict. So again, so they, in the adult state, they perfectly predict the age, but this is not aging. So this is a response to aging. And therefore, in certain, when we test interventions, often this clock does not work. So of course, this is just the beginning of separating kind of causal and defining causal inferences, but of course, it just needs to be further kind of studied in the field. And also, one more example is we developed this tool called ClockBase. Also, the same person, Albert Ng, developed this. Basically, we apply clocks, the most commonly used clocks currently, to the entire GEO database. So if there is any data that somebody deposited to GEO database and you're interested, you can just go to the website, and it actually has nice visualization tools, and basically just analyze yourself any of the data associated with age, predicted changes in biological age. All these clocks like Grimm age, pheno age, and Dunedin pace, and things like that, all predicted for all of the entire GEO database. So in the end, what is biological age? We think it's a level of age-related changes, such as damage accumulation, but also other changes. And again, so we refer typically to a reference population when we discuss biological age. So it applies to cells we could predict biological age of cells. I showed you clock application of the single cell clock. We could apply to tissues, to systems, and to the entire organisms. I would say that we do not fully understand how exactly biological age of individual cells and tissues interact with one another. This is also, we currently study this actively in the lab, but there is no full understanding. One point I'd like to make is actually integrative parameter. That's the beauty of the clock, biomarkers. It is kind of integrates all kinds of different information to approximate changes in biological age. It cannot accurately quantify it, but can nicely estimate it. Generally, it increases with chronological age, especially in the adult state, but not always. And I gave you a few examples where there are deviations. For example, in the NECMO rat, there's interesting changes compared to mortality, and also during development, all kinds of different patterns. So I think that it's distinct from measures of functional decline and mortality risk. It's related, but strictly speaking, it's not the same. Again, so this is a controversial issue that still needs to be discussed, and maybe we can discuss in this panel as well. So I also show that it fluctuates. It not only kind of steadily increases like chronological age, but it can be increased and accelerated, like, for example, by severe stress, and also can be reduced, for example, by parabiosis and other means. It also can maybe targeted with diets, drugs, and lifestyles. Just there's no time, but I guess I showed you some calorie restriction and a few other examples. But anyway, so this is also many labs study that. It applies to all organisms with non-renewable cells, to mammals. So I think all mammals age. In fact, maybe all vertebrates age, presumably. And it also may be estimated with aging biomarkers like clocks. And this is the beauty of the clocks. I think it's a true revolution in the field where previously we were not able to quantify aging. We used measures like oxidative damage, telomere shortening, which is just so inaccurate. For the first time, we are able to quantify, at least estimate in some way, biological age. And suddenly it opens like all kinds of different questions, how organs interact with one another, when does aging begin, and how could we actually manipulate biological age? So finally, that's it. So just the acknowledgement slide. On the left, this is members of my lab. And I showed you some examples of the work from Jesse Paganik, Alex Artishkovsky, Bohan Zhang, Madi Mokri, Albert Ying, and Wayne Mitchell. Other people are involved in other projects. And on the lower side is former lab members who also contributed to what I presented to you today. All of our studies are in collaboration. You can see many collaborators here, and Steve Horvath and Vera Andrei who are here, but also many other kind of nice collaborations. And we're also fortunate to have funding on the right side to support all of these studies. And thank you very much.
Well, thank you so much, Vadim. You knew it was going to be spectacular, and there you go. Now, we are out of time for that talk, but I will take at least one question, if there is a particularly urgent one. Anyone raise their hand? At the back there, if anyone has the roving mic, please take it up to there. Of course, we have the panel at the end, at which I will hope to be able to give some time for audience participation, but let's have, go ahead.
So, yeah, thank you, Vadim. He's gone. I think he's afraid of my question. You know what's coming. So, hi, Vadim. It's Pedro here. So, thank you for your talk. I mean, we discussed this by email already. So, but the question is, the problem I have is with your definition of biological age based on cellular molecular damage. So, the question is, how do we know cellular molecular damage is the cause of human aging? And I know that you must, so in the end, you on the deconvolution of epigenetic clocks, you also mentioned causal epigenetic signatures. So how do you know they're causal as well?
Well, of course, it's just a word, causal. So we know that the CPG sites that we identify in this kind of Mendelian randomization genetic approach associated with age-related changes, they kind of work to reduce, kind of accelerate aging associated with changes in mortality and changes with kind of disease states and other kind of functional functional changes. So we think it's actually causal in terms of the reducing biological age. And on the other hand, there are some CpG sites which kind of go in the opposite direction. So they essentially protect against those changes. So we call them causal. So, I mean, in fact, this whole approach, Mendelian randomization, is a genetic approach to identify causal changes. So that's exactly, we just apply this to the aging state. But in terms of the damage, of course, as I mentioned, there's no consensus in the field. So I expressed kind of my opinion on the topic, because we know that damage does accumulate. We see it, like, you know, in a simple way, like mutations. You know, every cell accumulates mutations. All these epigenetic changes. We see the dysregulation and, you know, transcription and translation. We see byproducts. We see increased number of chemical kind of species. So we kind of see it and also partially quantify through aging, through biomarkers of aging. And we published over the years. We published many studies where, for example, we introduced the damage to organisms, and it kind of reduces their lifespan. So we protect against damage, it increases lifespan. But ultimately, again, so it's there's no consensus. So I don't know what you think about aging. So whether it's probably not damage, maybe functional decline, or maybe changes in mortality. So I think this is some topic that just need to be discussed. I mean, because when we talk to each other, we say, what do you study aging? What do I study aging? But apparently we study different things. Mm-hmm. So we just need, like this particular panel, we just need to brainstorm and discuss and debate. Yeah.
Yeah, when I was choosing the title for this session, I was wondering whether we should have an entire day called The Elusive Concept of Biological Age, because there's so much to discuss.